Good. Hi, everybody. Thank you for joining our webinar today with the Center for Analytics, where we'll be hosting Dr. Joe Geraci from NetraMark, and we'll be presenting on their exciting work related to the Center for Analytics. Before we get started, I wanted to first begin with some acknowledgements of this land, its nature, and its people, in particular the Anishinaabe, the Mississaugas, the Wendat, and the Haudenosaunee. And every other nations, recorded or unrecorded, that have taken care of this land long before us. This is land which we must continue to work together on taking care of, as a group. So before we get started with the NetraMark presentation, I want to give a bit of background on the Center for Analytics and what we do. So let me get started on that, and then we'll pass it on to Dr. Joe Geraci for his presentation. At the end of the presentation from NetraMark, we'll also have a chance to open it up for everybody here to ask some questions and hear from Joe. So without further ado, a bit of background on OBI and the Center for Analytics. So at the Ontario Brain Institute, as you may or may not know, we're a research organization that helps the many groups across Ontario and Canada to collaborate on major neuroscience research projects. This spans a number of brain disorder areas. And as you can see here on these purple columns, so to speak, are these major brain areas where we have research groups collaborating across multiple sites to address major challenges in brain disorders, such as cerebral palsy, concussion, neurodevelopmental disorders, mental health, depression, and others. A key piece of this approach is to create what we call integrated discovery programs. And these programs span multiple sites, as I mentioned, and multiple disciplines and specialties, such as clinical, neuroimaging, genomics, molecular, wearables, and other types of data. They also work closely with patient communities, as well as industry partners. So we're really forming this integrated approach to addressing major brain disorders and undertaking important research in the areas. This creates a really important data opportunity because we now have these major programs collecting data across multiple sites and across major modalities. So it creates really a richness and sophistication of data being collected across patient and participant populations. Overall, we have a network of over 30 institutions that work together through these integrated discovery programs that are all connected to many of these institutions and collect the multimodal data that I mentioned earlier. These data come on a centralized platform that we call BrainCode. We now have over 1,000 users. These data, importantly, are standardized. They are also made available for sharing and collaboration across these sites through data standardization and the federation. And finally, we have capabilities for data linking and analysis that are really key for enabling these researchers to access the data and make use of the data. So here's a snapshot of the BrainCode portal as it stands relatively recently in May of 2025. We have a number of study data that are now collected on the platform. These are multimodal, as I mentioned. We enable data sharing within these networks that collaborate with each other, but also we enable sharing with third parties that can access these data and make new discoveries from the data that were potentially never even considered at the beginning when they were collected. We have common data elements that help identify commonalities across these cohorts. And so these elements are collected in a similar fashion across all these different brain conditions, which helps with the comparisons and analysis and asking of new questions. And we also enable that data linking, which I mentioned a bit earlier. This uses a fairly sophisticated method using homomorphic encryption, which preserves the privacy of the data, but we can link this research data with health administrative data that are hosted at other sites. So for example, the Institute for Clinical and Evaluative Sciences, ICES in Ontario. So overall, this does create a really fantastic data opportunity. We have a rich set of data being collected across these integrated discovery programs, including from POND, CPNet, EPLink, Cambine, Connect, and Andri, and new programs that are now highly active such as CALM and Transcendent, which are new integrated discovery programs supported by OBI.
Francis, you're not sharing anymore. I just wanted to let you know your screen's not sharing right now.
Is it,
It just switched to you. That's all.
let me see. Thanks.
It's
Apologize for that. I'm not sure what's
I
happening.
could still see it. There must be some other issue.
Okay. Can everybody see this?
yeah.
Okay.
Yeah.
Great, thank you. We are experiencing challenges. So we talked about the data opportunity. We have challenges which include developing skill sets that can actually work with these multimodal data. We also need to help the labs and the startups who are working with these data to accelerate their innovation. So accessing the right type of compute, accessing the right type of data platforms that have the security and privacy requirements, and the skill set that are required to use these modern types and secure compute infrastructure. We also need to support the translation of these innovations in the healthcare setting, but also in the population setting. So there are a number of gaps that are still present in our ecosystem that we need to address. And so this is where the creation of the Center for Analytics was really instrumental. Our goal is to really help unlock the future of brain health via neuroanalytics innovation and translation. And so we have these three major focus areas with the CFA. Developing neuroanalytic skills to support the creation of resources and expertise that can apply to this to analyzing and working with these complex data. We also have the opportunity to help with that acceleration of the technology development and the methods development through access to high quality data, access to advanced computing, and also access to a growing set of tools that are made available through the ecosystem. And ultimately helping with that translational aspect of making your technology available to patients, to physicians, to communities. So providing that support with the validation of the technology is a key area. Support with navigating the complexity of regulatory and the privacy landscape is another. And also helping, in some cases, startups or labs with that productization of their application. How do you actually bring an app that's been developed in the lab to the point where it can actually be used by a set of real world users, through an environment that is made available, for example, through the web or through a set of tooling that can be integrated within an existing ecosystem of tools or platforms. So these are major areas on which we focus on. And part of the approach to addressing this is to actually support projects such as the one that Joe will speak to you a little bit about through NetraMark, which we call analytics initiatives. So these initiatives are really aimed at developing skills using the multimodal data, leveraging advanced compute, developing new methods, and having a clear translational strategy. Ultimately, they can contribute the data back to the community, or even these tools that are being developed and methods that are being developed. And finally, we have a community of practice that's being formed through the Center for Analytics that we're really excited about. And we're starting to see the groups come together, share their knowledge and expertise, and to help solve problems that the community might be facing from a technical standpoint, from a practical or governance standpoint. So far, we've supported 10 analytics initiatives that really span a broad area of topics. Some focused on clinical trials optimization, for example, predicting outcomes, digital phenotyping, treatment optimization, even developing core tools for analytics or the analysis of multimodal data, or generating models for AI or machine learning training, and many other topics. And I think this is just a burgeoning landscape that we're seeing, but a lot of great opportunities to develop technology that are really based on data and analytics. So we have a number of resources that are available for everybody here and for the community in general. The methods tutorial that are created from these initiatives are being curated and hosted on our GitHub page on the left here. There's a QR code. We also have our webinars that are recorded, including this one, that are available through our YouTube channel with a QR code in the middle. And last but not least, we've just announced this week the call for the next cohort of analytics initiatives. And so you can learn more about that call on our website. And the link is incorrect, but the QR code is correct, and will take you to our webpage for the Center for Analytics on OBI's website. So I'll stop there. Thank you, everybody. And I wanted to introduce our presenter for today for the webinar, Dr. Joe Geraci. Joe is a mathematician, medical scientist, quantum machine learning specialist, and machine learning scientist. He's dedicated the last 12 years to medical science and the creation of next generation causal AI tools to help prevent the failure of clinical trials, discover new drugs, and create a new taxonomy of disease. Machine intelligence is a passion of his, and he utilizes his love for deep mathematics and medical science to go beyond standard methods to empower human experts to find answers for patients. So thank you, Joe, for joining today, and we're excited to have you. I'll pass it on to you.
Great. You can hear my screen and see me.
We can hear your screen and see you. Yeah.
Okay, great. It's great to be here. Let me mute my phone. Yeah, so thanks for having me on. And thanks for the support from OBI and CFA over the years. What I'm going to do today is actually just talk a bit about our company, a little bit about the technology, the uniqueness of our approach, what we're actually doing out in the market now for real sponsors and pharmaceutical companies, and to also talk about the project that the CFA has enabled through their funding. So without further ado, I founded the company back in 2017, 2018. I was a professor, and I was associated with Toronto General and Toronto Western Hospital and Princess Margaret Hospital for a while. And then I eventually came upon a mathematical paradigm that was very interesting that actually changed the course of my career. A lot of my colleagues went into drug discovery, and what I ended up doing was focusing more on trying to understand patient populations. And this came really from looking at oncology and neuroscience patient populations. There's a lot of heterogeneity. So when I was, when I was initially engaged in starting this company, what happened was I had to find a name for the company. And so Netra, I always loved this term because I did some work in Sanskrit and Hindu philosophy. And Netra is Sanskrit for eye, like beautiful eye, to see. And I was talking to my wife about biomarkers, and she blurted out NetraMark. And so that is how the story starts, right? So this ability to see deeper into patient populations with this mathematically augmented AI. Now, what you're seeing here with Clint Eastwood, this is an interpretation that our president, Josh Spiegel, he was starting to understand what I was doing, and so he came up with this image. And so let me explain to you what this represents. So the good, the bad, and the ugly, right? So what what we have to adhere to when we're looking at patient populations, especially in clinical trials, is the fact that obtaining patients for a clinical trial is very expensive. For example, a typical Alzheimer's trial, each patient costs about um, $40,000 US. And so these are very expensive. So the patients are not the data sets are not very large. And so a big part of my focus has been on creating methods that can actually attack these small data problems. And so the way we're going about doing this is you have to admit that there are parts of the population, because it doesn't your sample doesn't represent the distribution of the data in the wild or in the universe. And so what you end up having to do is you have to concede that you can't explain everybody. And it's very simple to understand. There's some patients that you have as representatives in this data set that don't have anybody in the data that looks like them. And so what we do is we use a technology that actually restructures the data in terms of we really understand this group of people and this is how you characterize them. We understand this group of people, this is how you characterize them, but we don't understand these people. There's not anyone who looks like these other patients. And what this does is it produces a superpower in a sense, and I'll show you what I mean, right? But the ultimate goal in doing all of this is to, you know, when I was introduced, talked about my interest in a new taxonomy of disease. The real truth is that without understanding the heterogeneity of these disorders, that you won't be able to properly figure out how to drug these populations. It's like trying to hit, 5 target boards with one dart being the drug. And so what we do, let me just continue. Hold on. Sorry, guys. So what we do then is we use this to our advantage by improving the visibility in these patient populations. So what a lot of people are doing out there now is they're trying to model placebo response, they're trying to model drug response, and then they take what they learn and they try to use that to help understand what's happening in their trial and how to mitigate failures or a higher placebo response. The problem is this can be very dangerous. And the reason that is because there are elements that influence placebo response that can influence drug response. And so, sorry, guys. And so so what we do instead is we actually attempt to model placebo non-response and treatment response simultaneously. Now think about what this means. So this good group, right? So this good group is essentially people that pre-randomization, if they ended up in the placebo arm, won't do so great, at least statistically. And if they end up in the drug arm, they will do better. And so the question is there a way to find that intersection, a way to characterize those patients? And the answer is yes, there is. And the same thing for the bad. The bad are basically people that would have a tendency towards placebo response or towards your competing drug, because that's how clinical trials work, and they won't do so great on your drug. And by going through this process, you end up with this great visibility into how to influence your trial. Now, this part of the slide on the left just shows you the sheer, like, monstrous number of combinations. But this is basically what we're talking about. We're talking about finding the right way to characterize these patients and in a way that will be robust, that won't be overfit. And the magic that we bring to this field in this regard is a novel long-range memory mechanism that allows variables to feel each other in a sense. It allows this paradigm of machine learning, which is not based on trees, not based on neural networks, because those things require quite a bit of data. But it allows the system to find these magic combinations. And I'm going to be talking a little bit more about that for the people in the audience who are interested in machine learning. But this is what becomes possible. Okay, so this is not what we deliver to our clients, but this is an example of the power of the core machine learning. And let me explain. And the power doesn't come for free. There's a very serious trade-off. So remember what I was saying, when you model these things, that it's very easy to overfit because the distributions are not complete, meaning not everybody in your patient data set has enough people in the data set for a statistic to become meaningful, right? Other groups there are, especially in clinical trials, because they do such a great job at controlling noise most of the time. So what happens here, so what we did here is OBI and Cambine allowed us to have access to their data, and we're going to be publishing some of these results, but just as a preview. What we were able to, so the task here was can you find a way to characterize SSRI response so that, doctors know whether or not they would be a good candidate for escitalopram and other SSRIs, or would they have to go into a different treatment? And so what we did is we found a way to characterize these patients, and we actually tried to model this data in a variety of different ways, and so did other people who got this data. And it's incredibly hard. And so if I can just bring your attention to the second column of the bottom table, you can see that traditional methods don't do very well. XGBoost actually started getting something around 65%. In our paper, we're going to report the sensitivity and specificity and all that stuff. But just here, just to illustrate a point of the power of allowing a system to get access to this new type of feature selection tool, this multimodal high dimensional feature selection. And what happens is by using just advice from Netra AI, by just saying, okay, these are the variables we're going to use, and these are the people we can't explain. What ends up happening is you get this massive boost in performance. I don't believe the SVM result, but the XGBoost actually does replicate. And so we get this excellent model that tells you whether someone will respond, will not respond, but more importantly, that the model can't predict a certain class of people. So if you were to use this model, for example, to determine whether someone was a responder or non-responder, you would get a lot of I don't knows. In fact, out of the 172 people in this data set, only 49 people can actually be characterized. But this makes sense because we're talking about major depression, and major depression is an extremely difficult disease to characterize. And in fact, we understand now in the psychiatric community that we need a new taxonomy. We understand that there's inflammatory reasons for people to have depression. There's situational reasons. There's brain network reasons, and more, right? There's comorbidities, etc. And so what we're seeing here is that the machine is very honest about what it can predict, and by doing that, it reveals a power. And then what we do is we actually utilize that power, right? So what we actually deliver to our clients is something different. But we go through that exercise because what it does is it allows us to understand the variables. And so we're able to take these trillions of combinations I was showing you in that slide, that big 10 to the 15 number, and we're able to reduce it down to a size so that you can explore the space in a more robust way. That's the magic here. We find these amazing bundles of variables that give you explanatory power, right? And so what this slide is showing, it's kind of an illustration we show to our clients where we wrangle their data. We go through analyzing the data in a binary sense, which is what I just showed you. It cracks open the data set. It reveals all these different subpopulations, and some of them are explainable and some are not, meaning some patients just don't have partners, right? No one looks like them, and so you can't make a statistic. And models that don't consider that will end up overfitting. But then what we do, and we don't do any of this stuff where we cut out the middle and we try to find an effect. It's nothing like that. But then what we do is we reduce the variable space. And then what we do is we start messing with the variables in a more intimate way. And because this has to be feasible, you can't use that many variables to characterize a patient. This is a very important point I want to make sure people understand. If I'm looking for someone who's over 6'4", who weighs less than 150 pounds, has this genetic variation, has blood pressure in this rate, et cetera, you're never going to find that person. So what we do is we take all of this machine learning, these models can be quite complex, and we filter them through and we simplify them so that we give our patients what we call personas. Okay. And this is an actual example from an Alzheimer's trial that we've been able to demonstrate actually has some efficacy through these personas. So let me explain. So let's look at persona one. So persona one consists of three variables: creatinine, concomitant meds, and potassium levels, right? Just as an example. So, and then there's an interval. So there's a range suggested by the AI, and then there's the actual range of the population. So you want creatinine to be on the low side. you want these people to never have taken or not to be on proton pump inhibitors. And you want their potassium to be on the higher side. And what happens is if you go out into the universe and you select these patients and you rerun a trial, what ends up happening is you get this winning p-value. You get this 0.01 with this great effect size. This is essentially what the system is doing. It's finding the ways to characterize the patient so that you can push your next trial in the right direction, right? I'll do the next persona as well. So here we have the ADAS-Cog word recall between 6 and 10. This is not very good. This is ADAS-Cog gets scored the other way. This means these are people who are having a lot of trouble with their memories. You want cholesterol to be lower, and you want their systolic blood pressure to be on the higher side. Not necessarily high, but on the higher side. And so what this represents is a group, a type of patient that actually has room to improve, but that has physical robustness. And so they end up responding to the drug. And in this way, by using previous data, you have this way to push forward and to influence your trial. And then what we actually deliver now, we've been working very hard at actually hacking large language models because they're not very good at dealing with these large context windows. And so what we've been doing is we actually now have what we internally call Dr. Netra, an LLM that can take these excavated um, insights and create reports. And it's getting better and better at it. It's actually quite beautiful. When you ask an LLM and you hold its hand to try to do this, it's a complete failure. I mean, in one of the trials we did, it found one patient that it thought it was reliably, good for the trial, but it's not very good. And if you want to be convinced of this, there's been papers written that these expert systems like Netra AI coming together with an LLM is quite powerful, and I'll talk a little bit about that. This is the way that pharma companies can use our technology, these results. So let me just take you through because it's interesting to understand how pharma companies operate and how they think. So protocol enrichment is what I've been just describing, right? You use a previous trial, you learn about what's driving the advantage of their drug. Every drug has an advantage. Pharma companies might not want to hear that. They might want to think, oh, this drug's going to treat everybody who has major depression, everybody who has Alzheimer's. It's not the way it works. We're very complex molecular machinery, and there's a lot of variance. But what you can do, though, is you can use these insights to push your trial into that right direction by recruiting intelligently based on what we learn after a lot of replication, of course. Covariate analysis. This is a way for these pharmaceutical companies to amend their protocols to say, hey, we have additional analyses we want to do, and the FDA is open to this. Everything here is open to the FDA, as long as things are done on our side pre-randomization, so there's no bleeding. TPP. So this is basically um, it's basically this one in market access give a way for the companies to rethink the strategy of their drug. It's very important to communicate to doctors that there is an advantage for the drug for specific people, especially if you're in a very competitive market. So some drugs, they're going up against many other drugs, and maybe there's an advantage with their drug, and our technology allows us to pull that advantage out and for them to um, speak very clearly to the regulator and to the insurance companies and to the doctors about that, especially in the United States where the majority of our customers are. Precision medicine, very clearly the way forward, especially for psychiatric disorders and brain disorders in general, because of the heterogeneity and complexity of the brain. What we're revealing is that there are specific ways to characterize the patient populations that will respond to these drugs. And a lot of drugs that have failed could have actually helped people. And that's what we've been finding over and over. And so this is some work that we have been doing. We've been very fortunate to be working with the NIH with some ketamine experts in major depression. And because of the CFA now, we actually have been building a pipeline to add to what we did in this paper, where we use scales and MRI, and in this paper, I think we have scales. And then we've added MRI as well, and then we're going to add MEG on top of that. We've done work in ALS where we just shatter the patient population and we allow our system to literally zoom in and out of these patient populations and figure out where are those weaknesses, biochemically that we can attack. And then we put out this archive paper that you can all look at. It's an interesting approach, actually dreamt up by our regulatory officer Luca Pani at NetraMark. And it's actually quite timely because what we're finding is that LLMs have certain advantages and other types of AI have other advantages. And when you pair these systems, you move towards what we call the modularity hypothesis, which is essentially, if you look at the brain, the way the brain evolved, we have occipital lobes, temporal lobes, dorsolateral prefrontal cortex, and they all have different modularity of functioning, right? They all come together to assemble, intelligent mammals or animals. And what we're finding here is this is the way AI is evolving as well. And when you pair something like Netra AI with a large language model, you get these amazing kind of expert system that excavates these subpopulations that the LLM can never do. And then you get the LLM that's able to translate it into clinical language. It's quite wonderful. Now, what are we doing with the CFA? So, thanks to Francis and the crew, what we've been doing is we undertook a project where we're trying to expand upon work that I actually did years ago when I first met the OBI, where I was an academic in Toronto. And so I've always been highly interested in graph theory and networks. In fact, with under the U.S. government's support, I was able to solve a problem in quantum computation where I used graph theory networks to build a bridge between these very difficult problems in statistical physics and quantum mechanics. And so graphs came naturally to me. And when I made the switch from mathematics over to medicine, the primary mathematical object that allows this bridge is the graph, all right? And I'll talk a little bit about that later. So in this example, what we want to do is we're building a tool that we're going to be able to share with the community that focuses on MEG, volumetric MRI, and fMRI. And so for MEG and fMRI, what we're doing is we're using these graph theory to actually create variables that you can run through your statistics algorithms, through your gradient boosting, whatever machine learning you want. And that's what this pipeline is going to be doing. So far, what we've done is we've made progress. Thanks for the computer, Francis. It's really nice. We've run our NIH, some NIH data through it, where we have MEG data. And now we finally have fMRI data from Cambine as of a few days ago. So I'm looking forward to doing that. That has about 254 patients, so very exciting. Ketamine was a small data set, but it was a real trial, right? I wish Cambine put a placebo group in their data, right? But in NIH, even though it's small, we were actually able, it was a crossover study with ketamine, and we had MEG. And so we're excited to see what comes out of this. But we've already, as I showed you in the previous slide, we already put out a paper where we show how we were able to characterize this ketamine response and also attack something called functional unblinding, which is what happens when you try to use psychedelics that have mind-blowing effects and you try to hide the placebo response. And that's another topic. If someone wants to ask me about it later, I'm happy to get into it. But anyway, this is what we're doing. And this is an overview that we've been putting together. And basically, what we have is these three modalities: MRI, MEG, and fMRI. But the MEG and fMRI allow us, and this started actually years ago with work I did with Jonathan Downer, who's an expert on treating depression with transcranial magnetic stimulation. And so we wrote a couple of papers where we use some graph theory to try to predict who will respond to RTMS. And so now, years later, what we're doing is we're building a tool that kind of automates this. And so what we do is we turn the temporal signals that we have in MEG and fMRI into graphical structures. Not very complex. There's a variety of correlation, methods that you can use to parcellate and then associate the parcellations of the brain and construct a graph. But the real magic is what you do with it after, right? So there's all these standard centrality metrics like closeness centrality, betweenness centrality, eigenvector centrality, which is what Google worked on, especially in the beginning when Google first came out. And so there's ways of reasoning about this and extracting variables. And so I'm excited to actually have this almost completely functioning, and now we're validating, and we're going to be allowing the community to have access to this. Now, I want to talk to you guys a bit about my approach in graph theory. So this image came about when I explained to an LLM how I see graphs, right? So as a mathematician, I have a weird approach to some of these things. And sometimes I do see colors. And But one of the most amazing things about networks is, like, as a mathematician, what's amazing is these complex structures they can be understood by just looking at local phenomenon, by looking at the edges and doing certain computations. You can actually start understanding the complexity. It's amazing. The other thing is that we're essentially like networks. Where our brains are networks, our biochemistry are networks. Oncology can be looked at very clearly through a graph theory perspective. I mean, it's actually quite amazing. And the other aspect of graphs that I love is that you can actually do a whole bunch of mathematics on these objects, which, you would imagine, oh, you can't do calculus on a graph, but you can, right? It's not trivial, but you can. But these graphical structures themselves become spaces for opening up a whole new understanding of these biological phenomenon, clinical phenomenon, and so forth. So I'm very excited by that. And this is an example of what's possible, right? You have these regions of interest, you create these graphs, and they can be quite complex. And they capture the temporality of the data as well, which is really powerful. And then what we do is we extract these things. Like I said, this tool that we're building, thanks to CFA, is going to allow people like you to run your data through it, and it will construct these networks, and it will construct these very simple tables that we know are favorable to do machine learning with. So imagine having a whole whack load of new variables to analyze your MEG or fMRI data with. So before I finish up, I just wanted to talk about something that we have at NetraMark that I'm quite proud of, that differentiates our efforts and what makes this all possible. And what the most important thing here is that we're able to construct models that are not overfit. And this is very interesting from the perspective of large language models, which are trained on billions of tokens. And so we used to have discussions about what would happen. Is this going to completely overfit? And so what I wanted to leave the audience with is something to look up and play with, which is this lottery ticket hypothesis. This is the way deep neural networks are able to take these billions of parameters and be trained in a robust way so that they can respond in non-contrived ways, in flexible ways that we're finding with these, chatbots like ChatGPT. And this lottery ticket hypothesis relies on the fact that there's so many subgraphs in a big network, and that by initiating the subgraphs randomly, allowing it to learn and throwing out substructures that don't do well, you end up pruning your neural network down to a highly functioning structure. And so we call this the lottery ticket hypothesis because those subgraphs become lottery tickets. Now, what we did at NetraMark is, instead of a parameter space, what we're under, because doctors need to know why they're making decisions, is explainability, but we also wanted this power. And so what we're doing is we're applying the lottery ticket hypothesis to the variable space itself. And what we've been able to show, it's actually quite surprising, is that when you try to scramble the variables on some of these data sets, and you get machine learning to to learn from these scrambled labels, which don't make any sense, the machine learning will inevitably force some model, which is just, as we know, BS. But what's interesting here in our experiments is actually our system suppresses, it literally suppresses the artificial data sets. So in this graph on the top right, what you're seeing is that bottom pink curve. That actually is the honest, the model with the actual honest labels. And you see how it's underneath everything. It means that this actually matches the biology of this cancer more closely than all of the other scrambled ones. So Netra kind of refuses to put up with these false labels, and it will be very honest with its findings. Last, I just want to end with some credit to the CFA group for helping us out. So I just wanted to talk briefly about how you've done that. So Christian Kumba is an applied mathematician and computer scientist who works at NetraMark. All right, a really gifted guy. We have this grant with CFA that was called Major Depression Multivariate Patient Response Characterization. And he's a highly qualified person who was given the opportunity to learn about and build graph theory applications in neuroimaging. So CFA has made it possible for us to complement Christian's already great background and make him more flexible in terms of his applications in the neurosciences. Dr. Matt Carlin has a doctorate in neuroscience, and he's been brought on specifically to help us with these neuroscience problems and to help us build this pipeline and to understand a few things better, right? So Matt and Christian have been great on this project. And then finally, uh, I'll just say that being a Canadian company is very tough. We don't have the same kind of structures that the US does when it comes to support. And so the journey's been very hard. And our customers are in the US. And with the current environment economically around the world, we Canadian companies do need a lot of support, and I love seeing that groups like OBI and CFA are doing that. The good news is that we're growing, and that we're the failures of machine learning in this space have been great for us, and so we're now starting to be captured, and we're getting more and more customers, and just being able to stay alive over the years, and some of that came with your help, and has been great. So, and basically, essentially, CFA is providing us with an opportunity to create new ways to better serve these pharmaceutical companies that go beyond our already, unique blend of mathematically augmented AI and our operational excellence. So, thank you. And so that ends my talk. I can go into other things, and I can take questions happily, Francis.
That's great. Thank you, Joe. Yeah, fantastic. Very rich presentation. A lot of innovative thinking. We have fascinating conversations always. Um, all we have 15 minutes to ask questions for Joe. So if anybody would like to ask a question, feel free to unmute and ask your question in the chat, or you can type it also in the chat box on Zoom. So I'll let anybody who'd like to ask questions for Joe jump in.
It's all been solved, Francis.
It's all been solved. I see Abby.
Sure. Yeah, I do have a question. Excellent presentation.
Thank you.
Joe, actually met you years ago. We both went through Creative Destruction Lab together.
That's right.
I founded BenchSci back then. I'm currently
Oh,
working as
amazing. Good. Yeah,
yeah,
that's great, man.
yeah,
Good.
David and the guys say hi. Actually working together again with my CTO, David. We're working on machine learning approaches and neuroimaging approaches to the use of ibogaine, psychedelic,
Excellent.
working with Ambio Life Sciences out of Mexico, treating about 100 patients a year, trying to understand how we can utilize that data set. I'm very curious about the statements you made around the functional unblinding aspects in relationship to psychedelics. Yeah, if you could speak on that aspect and how the NetraMark solutions might be deployed to solve
Excellent.
those issues.
Yeah,
Thank you.
that's great, Abby. Yeah, and thank you. Great to see and great to see your success. So, Abby, I want to say something first, and then I'll get into the functional unblinding. So, we um, we did this project with imaging, where we ended up with each patient had 800,000 uh, voxels, regions of interest. And so this was a data set that literally crushed the pharmaceutical company's efforts when they hired other teams for the machine learning. Not because just the massive amount of data, but because of the heterogeneity of the population, right? And what I wanted to say is if you and I offline, I'd love to talk to you about this, but just to tell you what happened with this is pretty amazing. So the scale they use is called the SNOT because they were interested in science, right? And in this particular subscale, it's called the EARF because it's the ear, our sinuses affect. Okay. Netra was able to go through this with this long range memory mechanism and pulled out voxels right next to the Eustachian tube. It was impossible for it to be a fluke. It literally pulled it out, explained it, and the doctors went, "Holy crap." So there's an advantage in looking at it from our paradigm just because of it's so difficult to find those feature bundles, Abby, that there's that aspect. Okay, now to answer your question about functional blinding, this is the way I think about it. And we're actually going to be writing a paper, and I invite you to actually review it even before we put it out. Okay. Because I find ibogaine a very interesting drug, except for some of the cardiac issues. But anyway, so
We have answers, but we'll talk.
good, good, good. So functional blinding to me looks like this. So what happened was I did a project where we were trying to like, oh, let's use AI to tell us maybe we put people on niacin so they get all itchy, put them on benzodiazepines, like try to mask, so that the people in the control group, we all came out, me and the physicians, and we're like, this is stupid. Psychedelics blow your brains out. Like they all know, right? Okay. So then I came back. One of the doctors said, Joe, can't you do some fancy math? And I'm like, oh, I don't know. So what I did is I figured out this, Abby. I used the personas. And I had this idea from quantum mechanics, and actually Christian, who's on the call that, and I was mentioning in the last thing, he's a good mathematician. He kind of get where I was going at. In quantum mechanics, there's this non-commutativity, right? That if you want to measure an object's momentum and its position, there's a limitation. So I was like, hey, you know what? Why don't I ask Netra to personify simple, very slick models, two to four variables of the people that were doing better on the placebo versus the people that were doing better on the drug. And then what I did is I came up with a simple mathematical, I mean, my team doesn't think it's simple, but it's simple, Abby, where you basically quantify how much of that difference is. And then you go to the regulator and say, listen, you can't, they're going to be unblinded, but the characterizations of the placebo responders and the drug responders are so different and the physicians can make sense of it and you can quantify it consistently. What do you think? And this is what we're working on now. So that's how we're attacking it, Abby, by comparing the personas into two groups to say, look, ibogaine responders are characterized in such a physiological way. And what we found actually, and I'll give you a little peek into our paper, is BMI was playing a big role in the placebo response, but nothing to do with the actual drug response, right? And the psychiatric characterizations that were coming out. So anyway, I think you're getting what I'm saying. That's the way we're attacking it. Quantum mechanics, loosely, right?
Excellent. Would love to follow up and chat with you further on this.
Yeah, please reach out. Yeah, find me at Dr. Joe at NetraMark or find me on LinkedIn. Yeah, thank you.
Perfect. Thank
Okay.
you again, Joel.
Thanks, Abby. Great question. Anybody else in the audience would like you to ask Joe a question? Joe, I was wondering if you had any sort of insights on, some techniques that, the community could use already to leverage relatively small sets of data, because we're all, many groups are working with ML and we're excited to develop technology that's based on new techniques, but the data are always available in terms of quantity or variety. And I know you put a lot of work into this, and refining your techniques to work with relatively small sets of data and still being able to find these relationships.
Yeah.
And there's some tools that you make available through your platform, but is there a pathway to getting there, to understanding that, and to maybe leveraging techniques that you're familiar with that folks can familiarize themselves with?
Yeah, no, it's a great question. It's something people can take away with them. So yeah, like NetraAI is based on a trade secret that involves some pretty strange mathematics, and maybe one day we will release that. But and from a NetraMark perspective, people should feel free. And I've done this for other groups. Actually, I've done it for a group inside one of your cohort, like inside the cohort, where they were having difficulty modeling, and we just asked NetraAI, "What would you do?" And we gave them the variables, and then they used gradient boosting, and they got a beautiful result. So I'm always open for stuff like that, guys. But in terms of just you're a practitioner and you're learning, I'll just give you some advice. The advice is this. Number one, our physicians and scientists don't have a clear picture of the heterogeneity of these disorders in brain diseases. We're not knocking the doctors. The doctors are important because of their clinical experience, and we're not knocking the scientists because they are exposing the etiological factors. So don't get me wrong. But when it comes to machine learning and getting robust models, you have to keep in mind that this stuff is very lumpy. So what you want to do is you want to push down the number of variables that you include in your models. And what you want to do is start off with regularization with logistic regression. Let it fail, but learn from it. And then move to random forest and gradient boosting. And don't just throw all your variables in. Try to intelligently try to find these variables. And even if it takes your computer 3 days, try to use wrapper methods and create bundles of variables that you can then experiment. It's not enough to just do fine tuning and parameter, hyper search. You need to use methods that are good for small data, like support vector machines, play with different kernels, logistic regression. And the ensemble tree methods are the best machine learning methods. No matter what people tell you about deep neural networks and all that, they're the best when it comes to doing really complex things like, language and understanding us and image, like convolutional neural networks, all that stuff. But when I worked at Equifax, I was part of their machine learning department. I kind of led how, the direction they were taking. I had an opportunity to do thousands of experiments with every technique out there. And those ensemble tree-based methods are extremely powerful, but you need to hold their hand with small data. And you need to not, you can't, it's very easy to fool yourself into thinking you have a model. What we've learned at NetraMark is that, I'll give you a concrete example. We took the old Katie data set about schizophrenia, and no matter how we tried, we couldn't enrich perfenazine, but we couldn't enrich olanzapine. And so, like, why? And it just came back to us, like a physician basically interpreted it, said, yeah, you can enrich because perfenazine is this like hammer, right? And the Netra is just being honest about it. And so you just have to realize that sometimes you just can't find a model that will replicate because of the nature of what you're trying to model. These are not pictures of dogs and cats, right? Yeah.
That's fantastic. Yeah, great tips. Yeah, start small, break it
Yeah.
apart, learn from the from the subset of data, from the incremental modeling, and also
And
understand that, yeah, sometimes it just doesn't work because
yeah, and learn how to interact with your LLM. Like give the data to an LLM, talk to it, right? It's going to screw up. You tell it, okay, run a logistic regression now, and then, right, do your own feature selection, go back and forth. But remember, an LLM, if you just watch an LLM play chess,
yeah.
it's amazing. You suddenly a rook appears out of nowhere. Like they're not good at these things, but if you hold its hand, you can go down somewhat down the road because it knows how to run these models and code quite well. Right. So that's the other side I'd say. Like learn how to play that game, especially for young people who are learning. It's a great time to be going into this now.
Mhm. I have another question if nobody else does before we leave, because we only have a few minutes left. But so far, this has been really amazing, Joe. I had a question maybe more on the sort of business side or that translational side.
Sure.
Working with pharmaceutical companies and companies who want to get a sense for the the validity of the algorithms, right, that you're proposing. What has been sort of their criteria? Like how much validation have you had to demonstrate to them to convince them that there's a pilot to be made here, like to actually run a pilot and get them on board and get them excited about what you're doing?
Yeah, this is the best question. This is the last 8 years of my life.
Right.
So, I'll say this, people like me and Abby and, who are like, when these companies that went early in this direction, we broke down a lot of barriers, but the ultimate barrier was for us was that, so I would show up and say, hey, we have this new method and it does this. And what happened, I'm going to be totally frank here and people might not like it. The machine learning people in these companies, Francis, act like an immune system against innovation. It's, and it's, it is, it was not just like a little bit, like literally illegal activity. I'm not going to get into, of course,
Mhm.
lying to their medical staff about like just,
Wow.
okay. Then what happened was failure started happening. So the doctors opened their door a bit and like, oh, maybe we should listen to Dr. Joe, maybe, right? Or maybe we should talk to in silico medicine. You know what I mean? It started opening up. And then what happened is eventually it's become more open. And what they really want to see is, all right, they pay, they get the results, and then they say, all right, we're not going to enrich right now. We're not going to do an enrichment based on your stuff, but what we're going to do is we're going to use what you have to influence our trial. We're going to use it to help us train our staff at the sites. And then what happens, the evidence started pouring in. And then the real magic is that we started amassing enough examples that we were able to show, look what happens at phase 2 to phase 3.
Right.
And for instance, the ultimate thing we're still waiting for is like, you know, phase 5, that company, NetraMark, whoever said to do this, right? Unlearn. No one's done it yet, but we have actually been able to demonstrate it. I can't talk about it though, but that is just a journey of dealing with that and then and opening up that happened. And actually, thanks to OpenAI, attitudes have changed.
Right. Attitudes have changed. Yeah.
And
Fantastic.
publications, you got to publish.
Yeah. Yeah.
Yeah.
Yeah. Amazing tips. Thanks a lot, Joe. Great presentation. Really appreciate the time today. Thank you, everybody, for joining the webinar today. This recording will be available online, so stay tuned. Links are available on our website and also on those QR codes I shared earlier, so don't forget. Yeah, thank you all for joining. Thanks, Joe. Really inspiring talk today. So
Yeah.
have a great day, everybody.
Thank you. Thank you, everybody.
Everyone.